Thank you, Andrea, for the really warm introduction, and thank you all for coming and being a part of this. Like she said, this is my PGM1-CDG journey, both as a patient and as a researcher from both sides of the bench. So, as Andrea said, I wasn't diagnosed with PGM1-CDG until I turned 18. Originally, I was diagnosed with Pierre-Robin sequence, which is largely characterized by the backward positioned tongue and the underdevelopment of the lower jaw, which ultimately leads to an airway obstruction. I was also diagnosed with a cleft palate. And I would love to go through my whole medical history from Pierre-Robin sequence to PGM1-CDG diagnosis, but as many families and researchers know, that would probably take up this whole weekend. So I would like to touch on two important milestones in my patient journey. The first was starting human growth hormone. Now, I didn't start human growth hormone until I was around the age of 14, and this was because I had no expectation of short stature or any hormonal deficiencies. This was just based on a stagnated growth chart. So on the left side, you can see that's around where I started my human growth hormone, and the red line is about when I finished it. So I was only on this for 2 to 2.5 years, and you can see a small upswing in the growth chart there, but not nearly as dramatic as you would expect with a growth hormone supplementation. And so this is not necessarily a cautionary tale, but more a call to action for kids who have CDG, where this could result in a short stature or growth abnormalities. I didn't start mine until my growth plates were mostly closed. So go early, meet with your endocrinologist, and see if human growth hormone is right for you. It was still impactful for me, and I'm very happy that I did it, but it could have done much more for me if I started it early. Here I have a few photos of starting my growth hormone. On the left was getting my blood samples to see if I was responsive, and on the right was my first night after taking the human growth hormone. My next milestone was sort of my penultimate experience as a patient, and this was a Lefort osteotomy. This was designed to correct the stagnated jaw growth and to fix the airway obstruction that started to occur more and more as I grew older. So as you can see in the image here, the goal is to sort of break the jaw in multiple places, move everything forwards to open your airway, and place everything with hardware so that the bone can reossify. This occurred around when I was at the age of 17. And here I have an x-ray of post my surgery, looking like what my parents called the bionic man. And here are just a few photos from my surgery day. After this surgery, I experienced some facial paralysis. And a year later, during a second opinion visit, I was prompted by the clinician to go and get a genetic screening because I had been screened at birth and at 3 years of age, but there had been no detection of PGM1-CDG because genetic sequencing technologies had not advanced far enough at that time. So at age 18, that's when I was finally diagnosed with PGM1-CDG, and I entered this wonderful community. A few years after that, like Andrea mentioned, when I began my master's program, I got in touch with Dr. Eva Morava, and she very kindly welcomed me into her lab, where I was able to conduct my thesis research on cardiomyocyte dysfunction, which I would now love to go into and tell you a little bit more about. So this is from skin cells to heart cells, using patient fibroblasts to create cardiomyocytes in a dish. So, the main treatment for PGM1-CDG currently is an oral galactose supplementation. And thankfully, this helps alleviate or improve many symptoms, but what it does not improve are the cardiac presentations. One of these is dilated cardiomyopathy, which is the enlarging and weakening of the heart, which ultimately results in an energy crisis. You can see over on the right side the difference between a normal healthy heart and a heart with dilated cardiomyopathy. And I think the quote at the bottom from Dr. Sunny Wong really emphasizes the importance of filling this research gap. She says, dilated cardiomyopathy is an early onset, life-threatening condition, and all deceased patients with PGM1-CDG succumbed as the result of complications arising from dilated cardiomyopathy. So again, you can see the importance of filling this research gap. So here I have a brief schematic flowchart of the experiment that we did during my thesis program. We start with obtaining a fibroblast sample from a PGM1 patient. These fibroblasts are cultured and then sent off to our collaborator, Dr. Kent Lai, who you'll hear from later today, at the University of Utah lab, the Lai lab. He converts these fibroblasts, reprograms them back into induced pluripotent stem cells, or iPSCs, and these are then sent back to us where we then differentiate these into induced cardiomyocytes. Afterwards, we plan to expose them to AAV9 gene therapy and then do a number of analytical experiments. However, due to a funding shakeup, which I'm sure many of the researchers are aware of, maybe too aware, and also my time constraints, we were only really able to focus on the preparatory stages of this experiment. So I'll be focusing on how we go from those skin cells to heart cells. Like I said, we start with harvesting patient fibroblasts, and this is done via a skin biopsy. Skin biopsies are normally performed during a routine clinical visit, and they are painless. We use a local anesthetic, and the single-use dermal biopsy punch is used to obtain the skin sample. Something that was incredible for me was during my research experience, I was actually able to donate my own skin sample via a skin biopsy, and my cell line is now one of the experimental cell lines in this project. Here I have a photo of my scar to prove it, and also a bit of a dramatized recreation of Dr. Morava performing my skin biopsy. Here we have an image under the light microscope of what those skin fibroblasts will look like once we have them in culture. Afterwards, like I said, we send them off to Dr. Kent Lai, where he reprograms the fibroblasts into the induced pluripotent stem cells using a viral vector. And I won't go too deeply into this because I know we've already spoken a little bit about viral vectors, but these are genetically designed to help induce transcription factors which sort of send these cells back to the beginning. Stem cells are the cells that lead to all cells. So we send those fibroblasts back into their original form of induced pluripotent stem cells. And here you can see the difference between our fibroblasts and our induced pluripotent stem cells, or iPSCs. Once we have our iPSCs, we then manipulate something called the Wnt pathway to induce the cardiomyocyte differentiation. Again, I won't go too deeply into this, but all you need to know is we use two things: cheer, which activates the pathway, and we leave them exposed to cheer for a few days. We then expose them to IWP2, which inhibits the pathway. And this turning on and turning off essentially tells these stem cells to go down the pathway to become heart muscle cells, or cardiomyocytes. Here we have our cardiomyocytes, and this is probably my favorite slide. I know Thomas showed you a brief portion of it yesterday, but you can actually see these induced cardiomyocytes contract or beat on the dish. And this was just incredible for me. I'm looking forward to eventually seeing my own heart cells beating just like this. So now that we have our heart cells, what do we do with them? Normally, when you're studying the heart of a human patient, something that you would do is an EKG or an electrocardiogram, and this is used mainly to study beat rate, electrical conductivity, and a number of other important markers. But since we're studying cells, we can't do a typical EKG. So we use something called the MEA, or the multi-electrode array analytical plates, which were also touched on yesterday. And these plates have electrodes on the bottom where electrical impulses are constantly sent through. And when the induced cardiomyocytes contract or beat, they actually act almost like a resistor in an electrical circuit. Those changes in electrical conduction can then be measured, where we can learn about the different states of these cardiomyocytes. And so what we found is that PGM1 deficient cardiomyocytes exhibit a reduced beat rate and contractility. So, it's my laser pointer. Up here, you can see the difference between a control and a PGM1 patient. Over the course of 5 seconds, a control beats, we have 4 beats for a control, and for a PGM1 patient, only 1. Here you can also see a significantly reduced number of heartbeats over time. And here, this is our contractility, or the intensity of our contraction, and however deep the spike is the intensity of the contraction. So you can see in the control, we've got a nice deep spike, whereas in PGM1, they're very shallow. So this is the weakening of the heart. Finally, I'd like to acknowledge everybody who was able to be a huge part of this project. Firstly, Dr. Ava Morava and Dr. Tomas Kozic, Dr. Graham Preston, who wasn't able to make it, and the rest of the team at the Mount Sinai Lab who supported me throughout this process. Dr. Kent Lai and the other members of our collaborators at the Lai Lab for all the work that they do. Dr. Andrew Edmondson for being both my practitioner and my advocate in getting me involved in this research, and Andrea Miller and the rest of the team at CDG CARE for welcoming me and allowing me to speak in front of you all today. And with that, I thank you all for listening.